Good afternoon, everyone. It's my great pleasure to be here to introduce our work about identifying transcript boundaries using deep convolutional neural fields. So this is a joint work with Zhenzhu and Sheng. So our work is motivated by RNA sequencing and transcript assembly. So RNA-seq is a widely used technology to identify and quantify expressed transcripts in a sample. So the typical protocol of RNA-seq is as follows. So we first prepare the samples with mRNA moleculars, and then we create the fragments of those moleculars, and then we reverse transcribe those fragments into cDNAs. And finally, we sequence those cDNAs to get sequencing reads. So the problem of reconstruct the full-length transcripts from the sequencing reads is called transcript assembly. So this is a very important problem. And how to do that? So usually the first step of a transcript assembly is to align all those reads to a reference genome. And by doing so, some of them will be spliced aligned. So like those red reads showed here. So those spliced reads are very helpful because they tell us the coordinates of introns of this gene. So we can identify coordinates of introns from the spliced reads. And then we identify the boundaries of the expressed transcripts. So like here, we can identify those two start boundary for the two transcripts, and also the two end boundary of the two transcripts. So those boundaries plus the coordinates of introns split the reference genome into partial exons or exons. So, and then we can build a splice graph based on those like boundaries and introns. In this graph, each vertex corresponds to a partial exon, and each edge corresponds to a junction. And we also add an artificial source vertex, which connects to all the start boundaries, and also add like artificial sink vertex, and add all the like 8 boundaries to the sink vertex. And we can then reconstruct the expressed transcripts from this splice graph. So from this pipeline, you can see it's very important to correctly identify coordinates of introns and the boundaries of the expressed transcripts. So among those two things, so it's very easy to identify the coordinates of introns because the signal is very strong. Like here, so for each coordinates of intron, there is a group of reads that align to the same position. So we can easily identify the coordinates of introns. But it's very hard to identify the boundaries of expressed transcripts because we don't have such strong signals like here. So in order to identify this like start boundary, the only information we have is that around this position, the coverage increase. But this signal is very weak and noisy. So the task of our work is to identify the boundaries of expressed transcripts from the read alignments. So this task is very challenging. I will give you 4 situations. We already seen this example. So if a new transcript starts in the middle of exon, then in order to identify this start boundary, the only information we have is the coverage increase around this position. But this information is pretty noisy and pretty weak because the coverage variation is like widely observed. The second case is that if a new transcript starts in the middle of intron, and there is some intron contamination that connects these two exons, then it's very likely that we will miss identifying this start boundary. So the third case, if in the middle of exon, there is a region that is not covered by any reads, then probably we might like report 2 false positive boundaries. Last, if there is like intergenetic contamination that connects 2 transcripts, then we might like misreporting those two boundaries. So this is a very really tough task. And limited effort has been focused on this problem. So some of the assemblers using a very simple rule called typical rule. So basically, this rule is to identify boundaries for those coordinates that the reads start, appear, or disappear. So this rule identifies those positions as like boundaries. So the shortcoming of this rule is that for all the four situations we just seen before, it will fail. Another better and sophisticated method is called MWU test, which uses the Mann-Whitney U-test and fold change comparison to identify positions that the read coverage significantly change. So despite that, this method still has very low accuracy because of its limited capability to handle noise and to model transitions. Our solution to this problem is a new method called like DeepBound. So first, given the reads alignment to a whole genome, we first cluster all the reads into different gene loci. And then for each gene locus, we further divided this gene locus into different independent instances using the coordinates of introns we can infer. So like in this example of gene locus, we have like 6 independent instances. And we will apply DeepBound to each gene, to each instance to infer boundaries. And so the method has like 3 parts. The first one is to prepare features, and then those features will be piped into our core model, which is a deep convolutional neural field. And this model will predict the probability for each position of being like start boundary, end boundary, or not a boundary. And those probabilities will be then used to infer the final boundaries. So this is the position-specific features. So for each position in an instance, we use the following features. So the most important thing is the read coverage at this position. So we also compute the p-value for each position that whether the one side of this position has significantly more reads than the other side of this position. So we use these 2 p-values as features. And we also compute the read coverage deviation within a window size. So we also use the sequencing information. So what nucleotide at this position is as a feature. So we encoded them as 4 binary features. And finally, we add another 2 binary features to indicate whether this position is a start or end position of the whole gene loci. Because the start and end position of the whole gene loci is very likely to be a start or end boundary. So we pipe all the features into our model, deep convolutional neural fields. So this model is a combination of 2 models. One is deep convolutional neural network, which is used to capture the long-range dependencies between different positions. And on top of that is a conditional random fields, which is used to model the transition between different labels, between different states. Like what's the transition probability between those 3 labels. So this model will give us the probability for each position, for each label. And with these probabilities, we use a simple greedy algorithm to infer the final boundaries. Like we iteratively identify the peaks of the probability and identify them as final boundaries. So one of the main challenges in this boundary detection is that the 3 labels are extremely imbalanced. Here is a simple like statistics. So because of that, if we use a simple objective function, then it will fail. Like if we use the accuracy, which is defined as the true positive plus true negative over all the samples, then we can just simply reporting everything as like middle state, which will give us accuracy of something like that. So this requires us to have a better objective function that is insensitive to imbalanced label distribution. So it turns out that the AUC, the area under the ISO curve, is such a good objective function. But the AUC is very hard to compute. So we then, to address that, we use the Wilcoxon rank sum statistic as a like unbiased estimator of the AUC score. And this statistic requires computing correct number of indicator functions, which is very inefficient in practice. So to address that, we use the polynomial Chebyshev approximation for the indicator function. And this end up with a linear form and can be solved very efficiently in practice. So this solves the label imbalance issue. So to train a deep neural network, we need many many training data. So to address that, we propose using simulations to train our model. So in particular, we use that many instances that is simulated from human species. So we will use experimental results to show that this model that is trained from simulation, human data, can also work very well on other species and on biological data sets. So the final model we have has 3 layers of neural network and 100 neurons in each layer, and the window size for the convolutional model is 75. So we then use both simulation datasets and biological datasets to compare our method with other 2 methods. So for simulation, we use Flux Simulator to simulate the ISCB, and we try 2 species, human and mouse, and we also try 2 different coverage, the 150 million reads and 15 million reads. And given the 6 RNA-seq, we use HiSet2 to align the reads to the reference genome, which gives us the reads alignment. And all the 3 methods will take the reads alignment as input and output a set of predicted start and end boundaries. So we see a predicted one is correct if it is within 50 base pair to a ground truth boundary. Because we use simulation, so we know the ground truth. And finally, we report precision and recall for each method. Here comes the result. So this is the result for human species and with high coverage simulation. And we can see our method, DeepCNF, marked blue here, significantly outperforms other 2 methods in terms of both precision and recall. And this is the result for human species with low coverage simulation. Again, you can see our method outperforms the other 2 methods. So this result shows the simulation for mouse species. And this is high coverage, and this is low coverage. You can observe the similar pattern with human simulation. So this shows that the model trained on human simulation data can also work pretty well on mouse species. So we also evaluate these 3 methods using some biological data sets. So we have 10 RNA-seq data, and we use the same pipeline, except that for the ground truth, we use all the known human transcripts, because for those 10 real biological RNA-seq data sets, we don't know the ground truth. And this is the result. Again, so our method is much better than the other 2 methods. So to conclude, we present a new method, DeepBound, to identify transcript boundaries using deep convolutional neural fields. And we use the AUC to resolve the label imbalance issue. And we propose using simulation to train our model. And the source code of DeepBound is available at GitHub. So we thank the two co-authors, Jin Ru from UCSD and Sheng from KAUST. And we thank the travel fellowship provided by ISCB. And thank you very much for your attention.
Thank you. Okay, any questions? I have one question. So now you have a tool that can indicate the initial side and the end side of transcripts. So have you thought of applying this to see whether, how much you can correct the current annotations, especially in genomes that are less complete or human even?
Yeah, that's what we are doing now. So we are like incorporating this part into a transcriptome assembler to see whether it can improve the performance of transcript assembly.
So you envision using this also to distinguish alternative initiation sites and alternative transcription end sites as well?
Right.
Okay. Okay, like from different transcripts, right? Like different isoforms.
Yeah, so we can distinguish like different starting positions or like ending positions.
Okay, thank you. Any?
What happens if you have transcripts going in the other direction, overlapping, things like that?
So I think if we use like strand-specific like RNA-seq sequencing, this is not a problem because we can distinguish the two like strands.
But if you don't have that?
If we if we don't have that, then we rely on the pattern of the splice site to try to distinguish which strand it is come from. Yeah. But those information is only available on those like splice trees. For the whole piece of read, which is not available. Yeah.
Okay, we still have time for a quick question. Yes, over there. Can you speak on the mic? Yeah, the mic.
Thank you. How effective was the solution of that imbalanced issue with approximation? What if there wasn't any solution with that thing, without any approximation?
I'm sorry?
What if you won't used any approximation of objective function? How this, what the performance will be without it?
So
was it effective or not?
So if we don't use, so it's mainly for the practice purpose. If you don't use the approximation, it's very hard to optimize.
Mm-hmm.
Yeah, so we couldn't try without approximation.
Okay, but there are a lot of other approaches of solving unbalanced problems. Have you tried it or not?
We didn't.
You don't need it. Okay, thank you.
Okay, so okay, can you mind to go to the mic, please? This will be the last question.
Yeah, just curious. Like, did you do any analysis? Because I didn't see like how many full-length transcripts you could find when you compare to reference genome, because that was the major problem with cufflinks.
Right.
So did you do any such thing? How much percentage of transcripts you could find?
Yeah, that's exactly what we are doing. So as we show, this is like a part of the whole pipeline of transcript assembly. So we are trying to incorporate this part into, actually we are now developing a new transcript assembler. We will combine those two to see whether it really improves.
No,
right?
what I mean is like you have like cufflinks when you have like, for example, like a lot of
right.
Reference transcripts, and then you could find like they couldn't really go to the full length and something just.
It's hard, to dig into their code to split this part out. So that's why in this paper we didn't show the final assembly results. Yeah.
Okay, thanks.
All right, thank you. Let's thank Mingfu again.
Thank you.